Okay, ready? Go down, down, down. Okay, to the right.
Oh, I see.
No, to the right, no, to the right, to the right, to the right, to the right. Yes, right there. Woo! What? Okay, sorry. Go back to your slides.
I am screen sharing.
Okay, good. Yeah, you're still screen sharing.
There we go.
Okay.
No. Oh, I have that on that. I turned it on.
Okay, yay! Technical challenges overcome. So, I know a lot of you know her, but I want to introduce her anyway. This is Brooke Rhead, and she was part of the Genome Browser group for 7 years. And in 2013, she decided to go back to grad school, and so she is now a PhD candidate at UC Berkeley. Yay! And she's in the computational biology program. Her advisor is Lisa Barcelos in the genetic epidemiology and genomics lab. And her current research focuses on the relationship between vitamin D and multiple sclerosis. And she'll be telling us about that and a lot more things as well.
Yes.
Yay, thank you for coming back.
Thank you, Anne. Thank you for having me down here. This is cool. I remember when Andy Pohl came back from Barcelona mid-PhD and talked to us, so I feel like I'm repeating that. But yeah, so I'll tell you about my current research, which is vitamin D and multiple sclerosis. But and that section of this talk is pretty much what I presented at ASHG. So Vina and Bob's not part of it. But now you get the revamp. But first, I'll just tell you about my program and my life in Berkeley and what that is like compared to here. So, that works. I'm at UC Berkeley now, obviously, and this is my building that I work in. It's Stanley Hall, and it's a very new, very pretty, nice building, and it reminds me a lot of E2, actually. And what's that? Yeah, this is the inside of it.
Wow.
Yeah.
Barbed wire?
Oh, Christmas decorations. Christmas decorations.
That's your hand here.
Just grad student. Christmas barbed wire.
Yeah, you got to every day crawl under it.
And that door right there is one of the many QB3 offices, so it is very similar to E2. And I think there's several different computational biology faculty and also administrative offices. So it's kind of housed in this building, which is nice. And then directly across the courtyard from Stanley Hall is this building, Evans Hall, which I actually have I share a little office in there too. So I feel like I'm kind of in both buildings. But to me, this is the Baskin engineering of Berkeley, because it's just this big cement blob directly across from my really lovely new pretty building. And I have to tell you that although the redwoods here are gorgeous, we are completely spoiled food-wise. So this is the campus, and this doesn't even have. I just searched for restaurants. This doesn't even have all the restaurants on campus, but this is the closest to my building, this north side. But pretty much 3 sides of the campus are just crawling with.
So, and where is your room?
My building is there. Wait, I'm not sure what I'm looking at. Yeah, about right there. So I end up walking over there and eating all the time. But we don't have a taco truck. There's an Asian food truck and a something else truck. But
You don't need trucks.
you don't need. But there are trucks, which is really confusing. But they're on South Side.
Rude Awakening.
Rude Awakening. Yeah. Lots of good expensive food, but also lots of good cheap food, because lots of students who don't tip ever, I don't think, go to all these places.
So Brooke, is Evans the old
LTS building?
It's the stats building, and
LTS building. that
Evans
I
Hall.
recommend.
Had a CDC 6400 in the basement.
what does that even mean?
It's a cookie.
And I think it's where the birthday units might have been for it.
What is
Yeah.
Art sorter.
Oh,
No, birthday Unix was in Evans Hall.
That's what I'm saying,
Yeah.
yeah. Evans.
No,
Yeah,
the other one up
sounds
in
right.
the corner, it's the engineering hall.
Oh,
They didn't do it in Evans.
it wasn't that,
No.
but you.
Yeah.
cool. Oh, and so then this is another thing that's not really that comparable. I took a picture because I thought it was kind of funny. On 420, which here is like the campus practically shuts down. That's, they drew a crowd. But like maybe there's 100 or 200 people tops. And like these people just kind of look confused. Like, why are those people just smoking? Like, right now? So that's just not on the same scale. Okay, so my actual program, it's the computational biology PhD program that I'm in. I kind of thought, like, so here I think we think of computational biology and bioinformatics as sort of synonymous, at least I did when I was here, and it's really not synonymous at Berkeley. It's the actual program is faculty. It's not like housed in any one department. It's faculty from all these bioengineering, statistics, biostats, 3 flavors of biology, computer science, of course, and public health, which my advisor's in public health. There are 40 faculty who identify, I guess, as being computational biology faculty. But 25-ish of them, at least according to the website right now, are in the core comp bio faculty group. So I think that means that they, well, I know one thing it means that they can, like, how they count on your qualifying exam or dissertation committee, like, but also they're just the people who like go to the retreat and are more involved in admissions and stuff like this. They usually get some kind of job like that. But that core group includes Kim and Scholander, who was David's student here once upon a time. And so I'm in the PhD program. And that started the year I went, 2013. So I'm the first cohort of people in it. There were 5 people in my cohort, and now we're up to 14. But before they had this PhD program, for a long time, they were having computational biology retreats and whatnot for a long time. But they had this designated emphasis that students who are like in some other program, so like one of these programs, they could declare that they like had an interest in computational biology, and it means they like take a few classes, and I'm not even sure exactly what else. But the alumni of that, there are 74 from that program, and that includes Ed Green, who is now here faculty. So here's my lab. It calls itself the Genetic Epidemiology and Genomic Laboratory. And here's my advisor, Lisa Barcelos. She's an immunologist. Her PhD is in immunology. And then she's also a public health person. She has a master in public health. This is our group as of Monday. It was a going away party for somebody. And so we kind of use any excuse to make food. So this is my advisor who brought in 2 waffle makers, and we had dueling waffle makers, like just during normal lab meeting, and like everybody brought toppings, and we had waffles. We've also had pancakes before, like brings in a griddle, and for the week before I did my qualifying exam, the lab meeting for the day was to do additional grilling of me and another person who was about to take her qualifying exam, and so we made grilled cheese sandwiches.
Nice.
So yeah, advisor is all about food, very Berkeley. Okay, so genetic epidemiology. What is genetic epidemiology? First of all, epidemiology is the study of the distribution and determinants of disease, which means who gets diseases and why. So I'm starting to now, I think when I went there, I didn't have in mind like, oh, I'm going to be a genetic epidemiologist or an epidemiologist. I just didn't know really what it was. But thankfully, we do rotations, and I found this lab and realized it really kind of fit my interests. We study in particular complex diseases, which means that it's diseases that are influenced by both genetic and environmental factors. And our focus is on autoimmune disease, so MS, which is very near and dear to my heart, and lupus and type 1 diabetes and rheumatoid arthritis. I've actually also worked on rheumatoid arthritis in this lab. So what this means is that now I do a lot of data analysis, and I mostly work in R. I usually either work in R or write bash scripts still, which is a skill in this lab that most people don't have, so I'm thankful for that. I make a lot of PCA plots. I worry about bias and confounding and study design. And most importantly, I am a genome browser user, which is something I put in my admissions essays, I think, that I wanted to go to switch sides. I wanted to go from being somebody who supports the genome browser to somebody who would actually use it. So I guess I have accomplished that goal. So speaking, what is that? We think there's a small child here.
It's a genome browser user. It's a cat.
So because I'm a genome browser user, I actually have two things that I want to ask for. I'm actually, I kind of forgot the fact that there's a suggestion box, and of course that I could just email people or whatever, but you don't get to these things, and so as I was getting ready, I was like, oh yeah, I should mention those things that we're always like wishing we had. So the first one, and now I'm really glad that Angie is listening. The first one is we would really, really, really love to have allele frequencies on the SNP details pages that aren't just a summary. Yeah. Okay, good. I'm not the only. I'm not like just an outlier. But yeah, population-specific ones, because that's we constantly need. Usually we are looking at a homogeneous population, and usually it's Caucasians, because that's who gets studied the most. But we're actually trying to branch out a little bit. But yeah, population-specific allele frequencies, and I know they must exist because I go to dbSNP to find them. But there's probably some complications. The other thing that I would really, really love to see is, remember the LD tracks? Yeah. These are back on HG18, I believe. Yeah. And it's, I think this is HapMap 2 or something, phase 2. Yeah. I'm not sure, but it's old and it's...
Angie has told me that the code is broken and somebody else wrote it.
Daryl.
Not supporting it anymore.
Remember Daryl put these up.
Yeah, but the original code
that
generates the data, yeah. not the display code.
Oh, the data.
Yeah.
Huh.
That generates the data.
That does the
LD calculations.
Ah, the LD calculations.
It's here too.
So like, this would be lovely. There
I
are.
have a friend who wants this too.
Yeah. Okay. Good. So there are places to go to get this information. Actually, but a lot of it seems like in around 2008, a lot of people got interested in like linkage disequilibrium, which is like how SNPs are inherited together, and the patterns. but nobody's maintained them, or like, I don't know. Okay.
Yes, I will put up through request for them. That was a big topic of interest in the meeting I was at recently, the genotype.
Yeah?
Special people are super interested.
Yes,
Yeah.
yes. Okay, good. So these two things would just make our lives so much
People
easier.
like this display? Is that kind of
It's
the best
the-
thing going?
Yeah, I think so. I think this came from HaploView, which is like
Okay.
how it started.
Nobody's
and
invented
as,
a better way to display this.
as far as I know. And then it would actually be really cool if you could get like, well, it's probably in the tables. I've never looked myself, but just to see like the D prime and R squared, like the measures of how frequently
Oh, yeah.
particular SNPs are inherited together. Like, I'm sure we could do something cool with that.
And she thinks she can get the LD values out of 1,000 genomes.
Yeah. Yeah, and like there's 1,000 genomes phase 3 now, which obviously you know because you just released this track. But there's 2,500 haplotypes in that. It's called 1,000 genomes, but it's more than that. So, anyway.
Okay, I diligently took that.
That's my plug. Thank you. And I still show people how to use the genome browser. Not nearly as much, but I do. Last April, I gave a seminar. This says, it's blurry, but it's core skills in computational biology. And down there it says navigating the genome browser. And I borrowed some slides from Pauline for that, but it was kind of crazy because this core skills in computational biology is something that somebody in my cohort made up. He just, he realized, because for the first year, there's some growing pains, and he's like, I wish somebody had, there was like some way for us to all get together and like learn about these basic sort of things that are really useful. There's not like a core class yet that really teaches you about some basic useful stuff if you're a computational biologist. And so he put together this series. Well, we wanted to just basically meet together, but to get somebody to pay for food. So he did that, but then, since we did that, we like advertised it better. He did the first one on principal components analysis, and then I did the second one on navigating the genome browser. People just came out of the woodwork to come to this. I was like, who are all you people? What are you, like, they barely fit into the room that we got for it. So, yeah. And then there's of course this molecular and genetic epidemiology and human health in the 21st century, which is a mouthful. This is the grad course that my advisor teaches, and I have taken it once, and I will be TAing it next semester. But last year, and I'm sure probably for the whole time I'm at Berkeley, she's going to have me do a genome browser lab. So, yeah. Oh yeah. Also, I'm in student government. Yeah, which I think is like kind of bizarre because this was also never a thing that I was like, oh, obviously I should get involved in student government. But so I'm in the graduate assembly, and the reason that I even got into it is we had this new PhD program, and we got an email came to our group saying, oh, you don't have any representatives in the graduate assembly. Like, you should have a representative. And we were like, oh, are we supposed to have elections or what? And we also basically just kind of looked around at each other and was like, do you want to do it? And I was like, I'll do it. And so I go to these graduate assembly meetings now. And we got this new room. You can see, or there's this, we used to meet in this old shaggy building, but just the last 2 months we got to move into the brand new student union, Eshelman Hall it's called. And so it's very official. It has this graduate assembly logo up on the wall, and there is a person at the front who has a gavel, and we follow Robert's Rules of Order. And on the way in, I noticed in this new building, because there's also the undergrad student government in there, and there's a sign on the way in that said Senate Chambers, and I was just like, who am I? Like, where am I? What am I doing? But it's actually been a really good experience. They have these once a month, like 4 or 5 hour long meetings, which are they're marathon meetings. And they do cater them, so that's nice. But I always walk out of these meetings feeling like grad students and students in general just have a lot of say about how the campus is run and how money gets spent. We have there's a budget group that we always have to spend a long time arguing about how, because I think that it gets money from student fees, and then they get to disperse it and decide how to spend it. And one of the ways that they spend it is on social activities, and they always, for the grad students, they have so much booze. Like every social activity is run on free booze. So, but they're concerned about housing and diversity and healthcare for student parents, and it's just like all manner of things that affect students, which I think is really cool. And I looked to see, I don't think there are any grad students in here right now, but I looked to see if UCSC has a similar thing, and there's a graduate student association, and their process for getting new people, if your department doesn't have a representative, you just show up, and then they take a vote at the end of the day, like, do you want to approve this person to be on the grad student association? So I have no idea if it's like as sprawling and big as the UC Berkeley one, but like grad students know they should do it. So, and they said like these are all the departments that don't have a rep right now. So computer engineering is on there, MCD biology is on there. So yeah, interesting. Yeah. And I go to conferences. I've been to 3 so far, 2 ASHGs and 1 rheumatology conference. But ASHG is the big one for my lab. Everybody goes. My advisor does not miss this conference. So if you go to ASHG, then you will see me there at least for 2 more years. Yeah, at least until I graduate. And so these pictures on this side are the this year's ASHG, which is where I gave this talk. So this is the actual work that I'm doing. Mendelian randomization provides evidence for a causal effect of low vitamin D on multiple sclerosis risk. So our data comes from Kaiser Permanente of Northern California. We do a lot. I think my advisor is she's somehow affiliated with them. We also have collaborators at UC San Francisco that we do work on other diseases with. And actually, we have, we're less involved, but we have people in Kaiser Permanente, Southern California, and they're doing a whole bunch on pediatric MS, so that's cool. Yeah. So, MS, if you don't know, is an immune-mediated disease affecting the central nervous system, and your immune system attacks the myelin sheath around the nerves in your brain. It's only in the central nervous system. I didn't realize that before. This leads to all manner of symptoms and disability, just anything that you can think of that it's important for a signal to be transmitted from your brain to the rest of the body. It can affect that. The average age of onset is 30, which makes it one of the leading causes of disability for young adults, and affects over 2.5 million people. And the risk factors for MS, the first is sex. It's 2 to 3 times more common in women than it is in men. More common in people of Northern European ancestry. And genetics definitely play a role. The published number right now is actually 110 MS-associated SNPs. they all have tiny, tiny effect sizes, but there's the group that is sort of responsible for putting this out is the International MS Genetics Consortium. And I think they have about 100,000 people in their genetics, you know, in their GWAS, basically. And like continually adding to that, but they're soon to be coming out with a paper that says they found more. Then there's the big one for this. So these all have tiny effect sizes. This has the biggest effect size by far. It's this gene HLA-DRB1. So that's in the MHC on chromosome 6, which is where you have the most diversity by far. There's like the number of alleles. This is allele for a whole gene, so like several, like a haplotype, basically, just several different SNPs that tend to be inherited together. But there's like several hundred alleles for this HLA-DRB1 gene. And yeah, this is the one that is associated highly with MS. There's also environmental factors. So this is not a complete list, but these are the big ones that are just like not in dispute at all. Tobacco smoke, Epstein-Barr virus, which is the one that causes mono, and living at an increased distance from the equator. And that has been noticed since the 50s. And that is what this latitude gradient is what led to the investigation of vitamin D as a risk factor.
Smoking good or bad for it?
Bad. Although,
I was going to say, yeah. Stop smoking and move to the equator.
Yeah, stop smoking and move to the equator. And if you move to the equator, like age makes a difference. If you move there before the age of 15, you take on the risk of that. Or conversely, if you move away from the equator. So yeah, so that's some indication that it's vitamin D is important early, maybe, probably. And what was I going to tell you? I don't know. I forgot. Okay, so low vitamin D is associated with MS, and we know association is not causation. So my whole project is to try to figure out if it's really causal. So studies, many different kinds of studies have shown the association. And a few of them make us think that it looks causal. So there have been a couple of prospective studies, so where they've gathered vitamin D, like serum vitamin D measures on a big, huge cohort of people. Like there was one that was many millions of people in the military, and then another one in Sweden, I think, that was more on the order of 150,000. But people who had higher serum vitamin D levels up front had reduced risk of MS. And then looking at people, there's something called clinically isolated syndrome now that gets diagnosed, where you have symptoms of MS, but it's the first symptoms you've had, and like really, in order to diagnose MS, you need multiple kinds of symptoms spaced far apart in time. But they know that a lot of the clinically isolated syndrome people will eventually go on to develop MS. And there's now FDA-approved drugs for, like, you have clinically isolated syndrome, they will start you on drugs already. And so in that group of people, low vitamin D predicts a higher MS activity and like faster progression. And then there's also there's a dose-response relationship between low vitamin D and high MS activity and progression, and you see that in a couple different studies, which it's just sort of, it's leaning causal when you see that dose response. It's really hard to show that anything is causing anything else in people, especially, and randomized controlled trials are the gold standard for this. But yeah, we haven't randomized a bunch of people, just like whole communities to low or higher vitamin D.
How many people, if you know, in the U.S. have have MS right now?
About 400,000 is the estimate.
That's quite a few.
Yeah, and it's higher. Yeah, there's definitely a gradient, more in northern climates. Also associated with Northern European ancestry, which tends to be in the northern climates too. So, yeah. Okay, so I'm using this technique called Mendelian randomization. This is also known as instrumental variable analysis, which apparently economists have been doing this kind of statistical trick for years. But biologists started doing it and named it Mendelian randomization. And this, I'm going to draw up this whole picture of a causal diagram. And so in these, like an arrow means like this thing causes that thing. So our whole question is, does low vitamin D lead to MS? And it could be that it's not causal at all, and that it's just this relationship is confounded. Like there's some other factor that is causing both things. It could be that it's actually going the other way. So you could imagine, and this is referred to as reverse causality. So you could imagine people with MS maybe don't go out in the sun as much, and they don't get as much vitamin D. Oh, and I didn't mention, in case anybody doesn't know this up front, but you get the most vitamin D in your system when UV radiation hits your skin and it gets synthesized. You can get it from dietary sources too, but it's not in very many things. It's in fatty fish. In the US and a lot of places, dairy products are fortified with vitamin D. I think this was started to prevent rickets. It's in some mushrooms. It's really not that much in your diet, and like the big amount comes from the sun. So just a problem with observational studies on this issue in general is that vitamin D, it's hard to measure for this relationship because it fluctuates with the time of year. When is it really necessary? Like what age is it really necessary to have a high enough vitamin D? And it fluctuates throughout your lifetime. So, you know, it would be great if we had multiple time points on people, but we usually don't. It's like a one-time measure for any like big study. So the trick is to use SNPs that are associated with vitamin D level as the measure of vitamin D. So instead, we don't have vitamin D measured on the people in our study. But if we use these SNPs, they aren't affected by sun exposure or diet or obesity. So it's not going to be, the relationship won't be confounded if we use the SNPs as the measure. Also, whether or not you have MS is highly unlikely to be determining what SNPs that are associated with vitamin D you have. So reverse causality isn't a problem with Mendelian randomization either. And it's called Mendelian randomization because the SNPs you get, this is determined during meiosis, and it's as if you are randomizing people to a lower or higher level of like lifetime exposure of vitamin D. So the first thing you need are SNPs that are associated with vitamin D. And there's a GWAS in 2010 that identified these 3 SNPs. And so these risk variants are the variants that are associated with lower vitamin D. And they're all in genes that make sense biologically. So this first one, these SNPs have also shown up, or at least these genes have shown up in more than one genome-wide association study for vitamin D, so that's good. So GC is, it codes for the protein, the vitamin D binding protein. It's called, so you see that everywhere, called DBP. There is also a gene called DBP, which I had to like go snooping around in the browser to figure this out because people get confused about that. But it does something else. So yeah, vitamin D binding protein just transports vitamin D everywhere. and you have vitamin D receptors in pretty much all cell types. So vitamin D can feasibly have an effect anywhere. Then this CYP2R1 gene converts vitamin D into the active form for the vitamin D receptor, so it's not active right when you first synthesize it. And then the other one that shows up is this DHCR7 gene, and that one's kind of weird because what that does is make cholesterol out of the same substrate that that vitamin D is made out of when UV radiation hits it. So like it maybe is competing away that substrate, and that's how it's associated.
So none of them are coding, huh?
Although, so the ones in the GWAS aren't coding, but the genome-wide association studies
They're
are
limited.
done. What's that?
Oh, well, they're just, if you have a GWAS and they were only to fit certain locations on the chip.
Yeah. So so we do. This isn't like sequenced data for every person. So the thought is that these are probably in LD with
They may not be
functional SNPs.
They're close to the causal.
They're close to the causal SNP. Yeah. So I know it kind of bothered me at first that we just like looked at things in introns and said, oh, that's the one. but no, it's like you need to do fine mapping in order to try and figure out what the real causal SNP is. But we just assume that these are next to the causal SNP. Stop working? Oh no. But this isn't working either.
Oh,
Oh
that's weird.
no. Right.
Yeah.
Exit show. Oh no!
What
No.
happened?
It's just
the laptop.
frozen-ish. Well, it's still timing it.
Yeah.
I wonder.
Yeah. Ooh, that might be.
Technology,
Am I
man.
still sharing?
Yeah. If you're sharing. Is she still sharing?
Yeah, but continue the slideshow.
Uh, and maybe just see if this will work.
Yes.
At least that works. Do you want to try this or?
I can stand back here. That's also fine.
What do we push it? Yeah.
Oh, wait.
Yeah, we take. First we.
All right.
Okay.
Very good.
Carry on. Okay. So the way we actually did this analysis is we constructed an instrumental variable, which just basically means that we took the number of risk alleles they had for each one of those genes, and for each person, so this is 0, 1, or 2 for each one of these, and then multiplied that by the beta value, so like the effect size from the GWAS where they came from. So that it was a single number for every person representing their vitamin D level. And then it's actually really straightforward. You just plop that into a logistic regression and to see if, like using that as the vitamin D measure to see if you can see an effect. And we controlled for a bunch of things that we had measured, which is nice. The big one to control for here is ancestry. Because when you think about that causal diagram, let's see actually. Oh, too many animations. Ancestry is something that could still confound this relationship, right? Because your ancestry determines what SNPs you get and has a lot to do with whether or not you get MS. So highly important to actually still control for that one. And so we did this in 2 study populations. Whoa, careful. And this is epidemiologists love tables like this. They call this table 1 always. It's just the characteristics of, yeah, like, they're like, oh, well, where's your table 1? And it's like, that's just always what table 1 is, the characteristics of your study population. So these are our people from Kaiser Permanente. We have about 1,000 cases, 9,000 controls. And, oh, I think I even have a little circle. So more females than males, as you would guess. You can see, so the rate of smoking is higher in the MS cases. Like they had 50%-ish of MS cases smoke versus 32% of controls, or that's ever having smoked or not. This HLA DRB 1501, this is whether or not you had that risk allele. So 53% of the MS cases have it, and only 27% of not. So it's kind of cool, you know, you're seeing like the things that you would expect to see. Also, obesity was a little bit higher in the MS cases, which obesity is what one of my lab mates studies. She has done a lot of work showing, she's like one of the only people who have studied this obesity and MS relationship, so that's cool. And then, so that was our first study population. And then we have collaborators in Sweden. So this is the Karolinska Institute, and another about 6,000 cases, 6,000 controls. They have similar-ish, more of their controls actually smoke. But, you know, basically here is pretty much the same. Their obesity rates were lower. But, yeah, so we had these 2 data sets.
Oh, can I ask?
Oh, yeah.
Am I misunderstanding that? Are there, you said there'd be, you expect more MS cases up there, females and males, on that first line in your table there?
So females to males, so yeah.
So there's more females there, right? Which would make sense. But what about the controls?
Oh, they were matched on sex and birth, or
okay.
like age. Yeah.
Okay.
Sorry. Good point. Good. Very astute. Paying attention.
Would this be called table 1B, or would it be called table 2?
In the manuscript, which is in preparation, it's all in a single table.
Okay.
Single table 1. Yeah, I don't know. There aren't specific names for the rest of the tables and features. It's just always table 1. Okay, and so for Mendelian randomization, there are a couple of really big assumptions that you make when you do this kind of analysis. The first one is that there is no relationship here with any potential confounding factor and the SNPs that we used for our instrumental variable. And this one is, at least for everything that we have measured, this is fairly easy to check. We just look to see if there's an association between this score that we made for everybody and then everything that we actually have measured, which was quite a few different things. And so no relationship. Good. The second one is a lot harder to actually prove. And that is, you're assuming that these vitamin D SNPs, so what we're thinking as vitamin D SNPs, you're assuming they're not just going, like, acting on MS and just causing MS some other way. And so, hard to really prove that, but we're happy that those 3 genes where the SNPs are found, they like have an obvious role in vitamin D level. So that's hopeful. And then there's a paper that came out a couple years ago where, because vitamin D is sort of a hot topic these days, it's getting implicated in a lot of immune diseases, autoimmune, and just anything that has to do with inflammation. So this paper looked at a big group of people who had vitamin D levels measured and looked at a whole bunch of other things that were measured in those people, basically, like blood pressure or whatever, and like looked for any association and didn't find any. So we think that this assumption is okay to make. And then here are our results. The punchline is that we did find a decreased risk of MS as that score increased in people. And so this is what I was maybe going to write up on the whiteboard. Odds ratios. We think in terms of odds ratios constantly, but I did not know what they were before I started. So I assume people don't know this, right? Let me just write up on the whiteboard. Chalk talk. And yes, I did just do this in the qualifying exam. So an odds ratio, and odds, which is just similar to risk, but you can't measure risk directly in a case control study. So just like the probability of getting something over 1 minus the probability, like odds in horse racing, I guess. But the odds ratio is the odds that you have MS given that you're exposed to something over the odds of MS given that you're not exposed, you're unexposed. And so if you have an odds ratio that's less than 1, that's protective. And then greater than 1 is a risk. And 1 just means they're not associated. Your exposure and MS are not associated.
And in this case, exposure would be low, having low vitamin D.
Let's see.
You're saying exposed to vitamin D.
Right.
Because your ratio is
Exposed
down.
to vitamin D. So
Because your ratio is less than 1.
Yep. So in fact, I flipped this table back and forth. I made it low and high, so I'm slightly confused. But yeah, so exposed to a good thing and it's less than 1, it is protective. So the odds ratio up there is 0.79 is what we found in Kaiser, in the Kaiser study. And 0.86 is what we found in the replication.
So is it less protected in Sweden because of the latitude?
I don't know,
There's
actually.
got an environment on top of the SNPs.
Maybe, but then we also have this confidence interval here. And so a confidence interval, this 95% confidence interval, is the way to think about it is...
Still less protected in Sweden.
Yeah,
The bottom end of the
it
range
is.
higher.
It is. It's less protective in Sweden. But they're kind of a wide-ish confidence interval. So a 95% confidence interval, this is just a statistical thing for like sampling error that you would expect. So it means like if you were to repeat this experiment many, many times, 95% of the time, the confidence interval that you calculate will contain like the true population statistic. So it's to give you an idea of how certain you are about that confidence interval that you calculated. So like, I mean, we're pretty close to 1 here, but it's statistically significant, 0.04, 0.03 p-value. And then we put them together in a meta-analysis, so analyzing them all as one, combining them into a single number. And the meta-analysis just basically it weights the 2 studies together, and it weighted the Sweden study a lot more heavily, I think, because there's less variation. They have like an equal number of cases and controls, and it's very lopsided in the Kaiser study. So that's why I'm like, I guess I wouldn't read way too much into it being less protective in Sweden, because I don't think we have like that concise of a measure. But you would think, but you think it makes sense that it's less protective?
Well, it does because of the latitude. Because the SNP is one thing, And then you layer on top of that the lack of exposure to sun at that latitude. So you would think that
It just has less to do with.
there's less vitamin D available to them.
Yeah. Yeah. Oh, and so I wrote that odds ratio up as exposed or unexposed, but this IV that we made is a continuous. It's from 0 to 1.62, like is your measure. So for every 1 unit increase in your vitamin D measure, that's your odds of having MS changes by 0.85 times. Yeah. So the conclusions are that we showed that, or at least it provides evidence that low vitamin D truly is a cause of MS. Reverse causation is pretty unlikely here. And importantly, we controlled for genetic ancestry. This effect estimate, unlike most observational studies, is unlikely to be confounded by unmeasured or unknown factors. And our results were replicated in an independent data set, which is always very satisfying and publishable. Just want to thank the people that are in my lab, my advisors, Lisa, other PhD and MPH students, and these two people actually do all the, they run all the genotyping checks, basically. So there's a wet lab associated with my lab. I just never set foot in it. And it's a very sort of production-y lab.
Does having vitamin D help suppress symptoms in adults that already have MS, or does it is it critical at some earlier stage of life?
So it remains to be seen. There have been 5 double-blind randomized controlled trials for supplementation for people who already have MS. And they were all, well, one showed a sort of promising measures, the number of lesions were a little bit lower on people who were randomized to receive vitamin D. But they were all basically inconclusive, and they were also all tiny. The biggest one was 60 people, which is kind of ridiculous. So this is really good fodder for pushing forward. We need bigger clinical trials, and also clinical trials on people who don't have MS. Though that would be harder, though. But because it's more than just MS that they think vitamin D is having an effect on. Yeah, that could be in the maybe near future, a bigger, whole communities would have to be randomized, I think, to receiving more or less vitamin D.
So,
sorry.
So how do they supplement it with vitamin D pills, and how well do you absorb that?
So it makes, so the measure that is used in all of these studies is not the active form of vitamin D. It's that 25-OHD that was probably in here. It does make that number go up, but it's not completely clear if that actually affects your level of active vitamin D. And yeah, so you absorb it. You absorb it pretty well. I've also seen a clinical trial where they put people with MS in tanning beds, which I thought was pretty interesting.
Yeah.
So then it would affect calcium levels, presumably?
Yep.
If you have better vitamin D, you can absorb more calcium.
Yeah. Yeah, I believe so.
Is anyone measuring calcium levels or anything?
I think calcium is a lot better established already.
Okay.
And so, but vitamin D is causes immune cells to move, to cross the blood-brain barrier, or I'm not sure which direction that goes. There's a lot of, and they're not sure if it's actually UV radiation still. It could be that having a direct effect. It shifts the balance between Th17 cells, which are highly inflammatory type immune cells, to Tregs, which are T-regulatory cells, which sort of calm everything down. Which, yeah, everybody seems to be interested in inflammation, so, yeah.
Do you go back to the p-values for a sec?
Sure.
And on the meta-analysis. Boy, that p-value dropped a lot, didn't it?
Yeah,
On my side.
because, yeah, you've got these nice big numbers. Yeah, that's the whole point of doing the meta-analysis. And usually a meta-analysis is often in like a big literature review paper where they put together a whole bunch of different studies over many years. But yeah, you can also just do 2 studies. Did you have a question?
No.
You did. Oh, same question. Same question. Yeah. That's what I've been up to. Thanks. Yay. And for anybody who's not grossed out, I have some extra slides of what happened to my head. So if you don't want to see any blood, then you don't need to stick around for that. But if there's time, we can also. Fair enough.
Yeah, we'll stop the podcast.
I've seen.
Oh, yeah.